I'm going to just show this movie.
Every single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur. To understand how enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitate. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell in every living creature on the planet.
I use this movie just as a way to give you a visual impression of what each folded protein is doing inside a cell, taking enzymes as an example. You should now be familiar with the idea of showing a structure using, in this case, a ball and stick arrangement, or using different visualization patterns. And you could see how water surrounds the protein, how in a single reaction, how the substrate enters the protein is modified by side chains, which are positioned exactly in a way to convert the substrate into its end product. And you also saw the role of water in this particular example, which is there in the active site to help in conversion of this enzyme. So effectively, I want you to think a lot about water and the properties of water. In 10th and 12th standard, maybe 8th standard, you learnt about the properties of water. And water is life. Think about water in terms of its effect on macromolecules and the roles it plays in physiology. Okay, these are some properties I have listed of water. I have focused a lot on hydrogen bonding. Do remember that in a cell, 70% of the cell by weight is water. And also shown over here are the dry weight. After you remove water, whatever is left, how much of protein is there, how much of RNA is there, how much of lipid is there, and so on and so forth. And here is a sort of a schematic over here showing you water molecules, which are in the characteristic red, white ball and stick arrangement, and the hydrogen bonding network they have in side chains. So also shown on the left over here are water molecules surrounding a globular protein structure. So I won't say any more about this. I don't really have the time. I'm going to spend the last 10 plus 10 minutes on just introducing lipids and carbohydrates, and I will not go beyond that for this course, simply because, as I said, it's an introductory course. Now, lipids are not as easy to classify as nucleic acids or proteins. They are wide ranging in the different forms they have. Just like the protein data bank, there is a map structural database which lists the different unique lipids which have been identified. Unlike proteins which are identified by sequencing, nucleic acids by sequencing, mass spec is usually a tool to identify different lipids of different molecular weights, different kinds. Lipids are made up of these blocks, which are shown, the two red ones and the blue one on the bottom. And what I want to focus on is the fact that there are many, many classifications of lipids. And this is from the MAPS database showing you the kinds of classifications like sphingolipids, sterol lipids, polyketides, so on and so forth. And over here is what we are going to focus on for the next 5 minutes, which is lipids in membranes. Now, lipids in membranes are the massive bulk of lipids inside the cell. But remember, lipids are there in fat droplets, they are an energy source. Lipids conjugate to proteins. Lipids are signaling molecules. So lipids are there in have broad functions in the cell. But in terms of volumes, in terms of number of lipids involved in any one function, is basically lipids are there in membranes. And this is something you have seen. For example, phospholipids have, we draw these head tail diagrams, simplistic diagrams. Head loves water, tail doesn't love water. And the tail part mostly contains of these repeating blocks, which are carbon, hydrogen, and oxygen, three atoms mostly, which are hydrophobic. Okay. And shown in this picture is the usual double membrane arrangement with the head, which is polar facing outside and inside the cell, where there is an aqueous environment, and the hydrophobic parts hiding from water inside the body. This is a sort of a schematic picture showing you a membrane, which is in green and light green over here, with a phospholipid, with its diagrammatically shown over here with the atoms, fatty acids in the tail, a glycerol group, a phosphate, and a choline in the head part. And you can see water outside the membrane, water inside the membrane, a protein embedded inside the membrane. So this is what the normal structure of any membrane looks like. Water on both sides, a double membrane in the middle, and many times proteins, especially membrane proteins, sitting slap bang in the middle of the membrane, allowing communication between the two sides of the outside and the inside part of the membrane. And these are just some schematic diagrams of different lipids of different classes with the carbon, hydrogen, oxygen groups over here, and the polar groups shown in red and blue on the right hand side, with these three being mostly involved in cell membranes. Okay, so we'll see a quick movie to introduce lipids.
Fats are an essential part of a healthy diet. They contribute to the taste and texture of foods, like the smoothness of guacamole and the flakiness of a croissant. Fats are also a major source of energy and a critical component of cells and tissues, and they also help absorb essential vitamins, and can be converted into other molecules like prostaglandins, which help cells communicate with each other. Fats have a 3-carbon backbone called glycerol, as well as fatty acid chains. The fatty acid chain is basically a string of carbon and hydrogen atoms. When an OH group from the glycerol molecule binds to a hydrogen from the fatty acid, an H2O or water molecule gets released, and the two molecules link up. If this happens once, the result is a monoglyceride. If it happens twice, it's a diglyceride, and 3 times makes a triglyceride. Now, there are various types of fatty acid chains, and one way to categorize them is by their length. In other words, how many carbons they have. Short chain fatty acids have 2 to 5 carbons, medium chain fatty acids have 6 to 12 carbons, and long chain fatty acids have 13 or more carbons. Fatty acid chains are also categorized by the bonds connecting the carbons in the chain. A single bond is just one bond between the carbon atoms. And when a fatty acid chain has only single bonds, it's called a saturated fatty acid, because it has as many hydrogen atoms as possible, or it's saturated with them. Triglycerides with saturated fatty acids are nice and straight, so they pack together really well. And as a result, they're usually solid at room temperature. And the longer the saturated fatty acid chain, the more likely it'll be solid at room temperature. Carbons can also have double bonds between them, though. And when a fatty acid has one or more double bonds, it's called an unsaturated fatty acid, because it's not saturated with hydrogen atoms. For every double bond, there are 2 fewer hydrogen atoms. Also, a double bond causes a kink in the molecule, so the triglycerides don't pack together as nicely as saturated fats. As a result, unsaturated fats are usually liquid at room temperature. Unsaturated fatty acids can be further classified, according to the number of their double bonds. Monounsaturated fatty acids are unsaturated fatty acids with just one double bond. Polyunsaturated fatty acids have 2 or more double bonds. Also, they can be classified according to their location as well. Since all these hydrogens can get kind of crazy looking, we'll just take them away for now. So another name for the methyl end is the omega end. And then we can count the number of carbons until the first double bond. Since this one is 3, it would be an omega-3 fatty acid. If the double bond is 6 carbons from the end, it's an omega-6. And if it's 9 carbons from the end, it's called omega-9. Now, to make things even easier when looking at these molecules, I'm just going to show the bonds. Alright, so omega-3s are usually polyunsaturated fatty acids and include alpha-linolenic acid, or ALA, eicosapentaenoic acid, or EPA, and docosahexaenoic acid, or DHA. EPA and DHA are marine sources of omega-3s. They're produced by microalgae and end up in the tissues of fish like anchovies, mackerel, salmon, and sardines. ALA is found in plants like flaxseed.
This is just to give you an idea of the structure of lipids and how to draw them. Okay. I'll also now end by introducing you to carbohydrates. I just want to emphasize on a few things. One is that carbohydrates can form complex structures resembling, for example, what you see in nucleic acids and proteins. This is a helical form of starch or amylose. And again, hydrogen bonds play a very important role in this particular polymer. Okay. Let's look at a quick movie for introduction to carbohydrates.
Carbohydrates include both simple sugars, which are little ring-shaped molecules made of carbon, hydrogen, and oxygen, either alone or in pairs, as well as more complex carbohydrates, which are formed when these rings link up together to make long chains. Carbohydrates provide us with calories, or energy, and simple sugars in particular play a lot of roles in our diet. They sweeten lemonade, balance out an acidic miso soup, fuel yeast in rising dough and alcohol, and help preserve jams and jellies. Now, sugars are found naturally in plants like fruits, vegetables, and grains, as well as animal products like milk and cheese. Added sugars are the sugars that get added to foods, like cereals, ketchup, energy bars, and even salad dressings. To be clear, even if the sugar being added comes from a natural source like sugarcane or honey, it's still considered an added sugar. In fact, a variety of ingredients listed on food labels might be sources of added sugars. some of which you're probably familiar with. Sugar actually refers to a family of molecules called saccharides. Monosaccharides, where mono means one, so one sugar molecule. Disaccharides, where di means two, so two sugar molecules linked together. Oligosaccharides, where oligo means a few, so it's 3 to 9 sugar molecules linked together. And polysaccharides, where poly means many, so it's 10 or more sugar molecules linked together. Glucose is the most important member of the sugar family, and it's a monosaccharide. It's one of the main sources of calories for the body, and is able to cross the blood-brain barrier and nourish the brain. Another monosaccharide is fructose, which is commonly found in honey, fruits, and root vegetables. Finally, there's the monosaccharide galactose, known as milk sugar. It's known as milk sugar because it's only found in nature when it links with glucose to form lactose, a disaccharide found in the milk of mammals, which includes cow milk as well as human breast milk. Sucrose, or table sugar, is another disaccharide, and it's formed when fructose links up with glucose. Sucrose is found in various fruits and vegetables, with sugar cane and sugar beets having the highest quantities. Maltose is another disaccharide, and this one is two glucose molecules linked together, and it's found in molasses, which can be used as a substrate to ferment beer. Sugars, like fructose, for example, are most always found in combination with other sugars, and the combinations can be pretty different, even in seemingly similar foods.
So I'm going to stop this over here, and again I'll ask you to see the rest of this movie. So what I want to end is to emphasize that carbohydrates can form large polymers. Shown on the left are examples of 4 such polymers having the bonds which were shown in the short movie I showed you. And I'll take the example of hyaluronate, which has about 50,000 repeating units of this disaccharide. And this particular molecule and other molecules also are found on the surface of most cells. So the surface of cells are decorated with polysaccharides. Okay, it's literally it's like looking at a forest from down from a helicopter. You will see the surface of the cell is pretty much like the land. You cannot see the land at all. You only see the carbohydrates and you only see the protein. There's a huge amount of crowding at the level of the surface of the cell, with proteins and carbohydrates dominating the landscape of whichever entity is trying to enter the cell from the outside. So this is my last slide. I also want to leave you with the idea that simple carbohydrates and the polymers of carbohydrates, which you have seen, can conjugate to lipids and they can conjugate to proteins. Okay, and these conjugations between these 3 macromolecules, lipids, proteins, carbohydrates, have very important functional roles. And very obviously, looking from a central dogma viewpoint, these conjugations happen post-translationally. So DNA doesn't directly code for the conjugation of a carbohydrate or a lipid to a protein, but the unique sequence of amino acids has motifs which lead to conjugation of these other macromolecules to proteins. And by conjugation between these different macromolecules, you create functional and structural entities, which are very, very different from the simple idea of proteins, lipids, or even carbohydrates. And on the right-hand side, and this is the last thing I'm going to say, is a structure of the plasma membrane, with the bluish part on the left being the inside, showing you the polymers of actin filaments, which dominate the landscape of the inside of the cell. Membrane proteins like integrin sitting in the double membrane, which is made up of lipids. And as I said, a forest of carbohydrates and proteins on the surface of the cell, which effectively are pointing outwards. And any molecule coming to enter the cell or interact with the cell will first face this forest on its way to the cell. So I'll stop over here and take any questions.
Sir, what does the lipids exactly do?
So this is what I said, right? Lipids do many things. They are the structural entity of membranes. So the first thing which you will always say is that they are the membranes which are very important to compartmentalize cells, because unless you compartmentalize cells, different enzymatic reactions will mix with each other. So if you want a compartment inside a cell which is at low pH, for example, to degrade proteins, you cannot afford to have pH of 3 inside the entire cell, because the cell will not be able to function. So membranes will act as a sort of a barrier, keeping this compartment separate from everywhere else. Nucleic acids are themselves in a large compartment, a double membrane compartment called as the nucleus. So membrane proteins are very important. Now, lipids can also act as storage material, which can be used for making energy. Carbohydrates can be converted into lipids. Lipids can also function as signaling entities. So they are, means if you read a textbook or a review on lipids, there will be 40, 50 different things at least which lipids can do inside the cell.